Now, next up, we have Dr. Helen Liu. Dr. Helen Liu is the Percy and Vida Hudson Professor of Biomedical Engineering and Professor of Dental and Craniofacial Engineering, is the director of the Biomaterials and Interface Tissue Engineering Laboratory, and is also the Senior Vice Dean for Faculty Affairs and Advancement here at Columbia. I'll just hand it over now to Dr. Liu to tell us about regenerative materials for health and beyond.
Thank you, Jose, and thank you, Cameron and Parissa, for this wonderful organization of this meeting. And again, adding my welcome to all of you. And I think seeing all of you guys here today really brings to bear why we are professors and what life was like as a graduate student applying to PhD programs. So again, welcome, and I look forward to your comments and feedback. So in my lab, we're interested in integration of different tissues. So it's not just a regeneration of single tissues, but rather in the body, just like we talked about the collaborative spirit that Paul mentioned, tissues collaborate together, right, to form organs so that you can actually have function. So in my laboratory for the past 20 years, we have been interested in how tissues integrate with each other, how cells collaborate with each other. And these are manifested in the orthopaedic system as what we call interfaces. So we're familiar with interfaces, for those of you who have worked in biomaterials, is the graft and the host tissue, right? That's what we call the homotypic interface. And the heterotypic interface are interfaces between soft and hard tissues, whether it's cartilage, tendons, which connect muscle to bone, ligaments, which connect bone to bone. So these transitions are actually mechanically intricate in many ways and compositionally different, with a very gradation of properties that give manifest to these complex structures that enables functionality. So in the lab, we have many projects, and today I'm just going to focus on one, the cartilage to bone integration, or cartilage integration in general. So how, have you ever wondered how does cartilage connect to bone? And it connects to bones through a layer of, we know cartilage is not calcified, doesn't have mineral. So between cartilage and bone, what we have is a calcified cartilage, which probably makes sense, right? Something that's sort of in between. And this transition is very, very thin. So high fidelity, about 200 to 300 microns in humans. And it has a lot of job to do. So mechanically, it's basically, first of all, it's a barrier, okay, for vasculature to be able to go up to the avascular tissue. And then secondly, it allows the mitigation of stress concentrations. So you can go from soft to semi-hard to hard tissue, okay? So the challenge here is really how do you regenerate this? I put here results from a study from more than 20 years ago, probably, I don't know, before you guys were born, probably some of you, right? So that actually what they did, those of you who work in wet labs, you know about millipore filters that we use to filtrate stuff. So what this group did that in Switzerland, that they put a millipore filter. It's not even any fancy biomaterial, but millipore filter is actually pretty fancy material. So you put it there, and then in between, they create a supercritical size defect for cartilage, drilled it down to the bone. Then they put this millipore filter down there. And what they say that on the top, if you don't have a filter, you have bone ingrowth. So this barrier is not maintained. If you just have a filter, it prevents the ectopic calcification that we see. So the analogy is really like the Great Wall of China, right? You think about it, think about walls. So this is a wall. So how do you build this wall? Great Wall of China is very big, and this wall is very thin. So how do you build that wall? And it's really was an interesting challenge for integrating tissues. So as those of you who work in biomaterials, we start with ideal design criteria that's actually manifested through an understanding of structure-function relationship at these critical junctions. Okay, then you look at your arsenal as a biomaterial designer. Is that what kind of material do you use? So we tested degradable materials and non-degradable materials. And then because it's a composite, it has some little properties similar to bone and similar to soft tissue. So you bring a ceramic, a nanoparticle in, and then you talk about, does the particle size make a difference? You know, how much you put in, does it make a difference? And then who's going to do the work? It's the cells. Okay, what cells do you use? Which cells can you use, and what is clinically relevant, right? So then you have a bunch of sort of outcomes that standardize across all these variables that you test. So in many ways, this is sort of the standard approach for tissue engineering and biomaterial design. So I'll just highlight some of the studies here because fundamentally what was really interesting, and this is all of you who love nanomaterials, I love nanomaterials too, we found that, you know, the cells, these chondrocytes who make cartilage, don't really like nanomaterials as much as they like micromaterials, okay? And the reason for that is that they become much more hypertrophic when you give micron-sized ceramics because that's what they're used to, okay? So at the calcified cartilage transition, they're actually high-density cluster of mineral. That's how the barrier is actually formed. And then we also look at composition. Calcium phosphate is a very, very delicate, intricate ceramic material. So in the body, you have, during development, there's a whole range of calcium to phosphate ratios. Okay? And you can see the calcium phosphate ratio that was tested, 1.35 versus 1.41. It was statistically different. And we got a different response. So 1.41 got you a more hypertrophic phenotype than the lower one. So they can sense composition as well. Then cartilage integration has been traditionally challenging because you have to do both cartilage to bone integration as well to cartilage integration. So cartilage is avascular, so it doesn't heal. So you see that zone of death. For those of you who've done a live dead, you know that there's a gap right there. There's a zone of red cells. That's bad sign. The cells die. Okay, this is just for allografts, which is a gold standard for tissue engineering, right? So how can we actually help that gap to be bridged? So we developed a growth factor release device, and IGF-1 is actually a chemotactin, so it tracks cells. So idea is, can you take cells from the host and encourage them to come over and actually rebuild the interface? So this is an in vivo testing, in vitro testing, and you can see that the control group, no mesh. The middle group, just mesh without the chemotactic factor. So you can see it in the second panel. There's some stuff growing there, right? And the staying red positive is for proteoglycans, which is found in cartilage. And when you have a IGF chemotactant, that recruitment is much faster. Okay, so the mesh provides a place for them to go to and make tissue on, and then with IGF-1, it does a little faster. Okay, so this is overall summary of the approach that, you know, our goal is to design a system. So, you know, in terms of, we don't make cartilage grafts, okay, but what we do is that if you have a cartilage graft of interest, we can help you integrate, both with the host cartilage and the underlying cartilage. So this design has been tested in, just finished in a rabbit study, so we're hoping to move it to a large animal study. So what I have showcased here is really an application in terms of how biomaterials can be intentionally designed and scaffolds can be used to match what's needed for cell behavior. But fundamentally for biomaterials research, you're really thinking about cell-material interactions, right? A cell is like, what I say, like a super bright PhD student, right? You have your idea, you want to do something, and then how do we give you the resources to do it? And that's how you design your scaffold system. So these design cues come from nature. So if you look at collagen fiber, right, it starts at the nanoscale, and then it assembles itself into tendons or ligaments, right? So it's across multiple length scales. And we can present materials at multiple length scales. So what is the right length scale to present? And this is a chick tendon development TEM analysis was done by the Fresovers group in Rutgers. They were the expert in really looking at development of soft tissues. And you can see that the 10-day postnatal versus 1-day postnatal, your fiber diameter is increasing. Okay, so the body builds it up, right? So one of the studies that we looked at is, does fiber diameter matter? So again, we presented nano, micro, and nano 1 is like about 300, nano 2 is about 600 nanometers, and micro is about 2 microns. And the cells can tell the difference between them. And in this case, again, nano did not triumph. But what was really interesting is that when the cells see a nanofiber, they see an immature matrix. So they proliferate more. But when they see a microfiber, they're like, oh, this is big enough. And then they actually express the phenotype instead. So they can sense these differences as those lenses. These are actually the scales that they work in, right? That's how the cell interactions come in. And with recent understanding of immunology, and biomaterials are taking a whole new look at immune modulation, right? So can you design materials, instead of just saying, okay, we're going to have that acute immune response, can we just actually attenuate that immune response, right? So because tissues, when you can either have scar tissue for repair, or you can actually have true healing. Right? This is particularly important for tendons and ligaments because they are anisotropic materials. So you can design platforms for you to interrogate that. So that's another big interest in the lab. So depending, you have aligned or unaligned fibers. This is a paper we published more than almost 20 years ago that actually looked at effect of alignment and how it dictates cell response. So this was a no-brainer, right? If you give them a template, they're going to make the tissue based on that template. But what's really more interesting is that we know diameter matters, we know alignment matters. If you put them together, which one is more important? And the answer is that alignment is more important, okay, in driving that desired cell response. Okay. But if you want to drive the desired response of stem cells, you have to add another factor, mechanical loading. Okay. For phenotypic cells, alignment is the driver. But for stem cells, you have to do two factors. So we are also more recently we looked at making ECM platforms that are more physiologically relevant. So this is a collagen-based ECM structure that you can use to interrogate also the diameter and the alignment effects. Okay, so far what I have told you is about regenerative materials that we implant and has regenerative purposes. Okay, so for the next couple of slides, I just want to sort of shift a little bit to think about regenerative materials, and that's why it was sort of unique to be in New York and to be at Columbia. So we were using alginate. How many people have worked with alginate before? Wow, quite a few. Awesome. So it is a material that we use to test cartilage regeneration. Okay, so about 7 or 8 years ago, a faculty member at Fashion Institute of Technology approached me and said, "Hey, I heard that you're working with alginate. Would you be possibly interested in working with us, develop an alginate fiber?" I'm like, okay, but why? So what they were interested in was actually making fashion textiles. Okay, but why alginate versus polyester? Polyester doesn't degrade and contributes to the plastic pollution. Well, alginate does. Okay, so can you work with this biopolymer to make it strong enough so that we can knit a sweater? Or a vest, as my collaborator Tian Shiro is wearing that vest. Okay, and what was really interesting that the three Fashion Institute of Technology students started a startup. They called it Algae Knit to start, a very cute name, and now it's called Keo Labs because they raised enough and moved to North Carolina to build it to type. So they debuted their stuff 2023 at the Stella McCartney Paris Fashion Week, okay? And then they just went commercial, retail 2024. Like the retail to me is more interesting because you can actually buy that now online, using this fiber. Because this is what they were looking for is for a regenerative material that have a circular life cycle. So a regenerative can have different interpretations in different disciplines. And this is something that you will not be able to do anywhere else except in New York, because the Fashion Institute of Technology, which is the #1 fashion institute in the world, is actually in New York City. So these collaborations also is very humbling, because you heard about the human trials that Professor Sun was working on. And I worked on a ligament more than 25 years ago, and it's just going to FDA. And this project was 2017. and now they're on the market. Okay, so we get our satisfaction in multiple ways. But anyway, so in conclusion, this is sort of given rise to a new center. It's called the Gotham Foundry. Gotham, for New York City, and it's a material innovation hub for sustainable materials. And the idea is to create an open source, open door, and open access sustainable material innovations hub for people who love materials and who want to work with materials. And because building with biology, how do you go from biomimetic to bio-inspired to beyond biology, right? And you can see these materials made by cells. We are made by cells. You think about it, right? And these are things that are made by cells and can be made by cells for beyond biology and for biology. And so, if you're interested, there's a QR code for getting involved. And I just want to conclude on this video. You can also cook your food while you're printing it. This is how did they know? And it's regenerative. It was degradable at the end of its life. All right. Thank you for your attention.